Good morning, Jamie. Just calling to check in to see what you've been up to this week at the Horniman.
Yeah, good, good. So this week I've travelled across London. I've been working at the other side of London at the Natural History Museum in London, and I just started a position with them on top of my job at the Horniman as a science associate, and they're giving me access to their amazing lab facilities they have over here. So what I've been working on this week is documenting the embryo stages, the cellular changes of the embryo from fertilization right the way through to settlement of the coral. So during a coral spawn that we had in December last year, I collected specimens throughout that sort of 4-day process from the moment that the acropora released their eggs and sperm right the way through 4 days later to the planula larvae settling. And so I collected those specimens, preserved them, which is kind of fixed them in a state of time. And then and they're preserved forever now, you know. And then this week what I've been doing is then going through a whole bunch of stages that I needed to get those specimens preserved, so I can put them under this thing, the electron microscope, which is just super cool. It's an amazing, amazing piece of kit.
And what I've got next to me is a whole bunch of samples that I took from one of the coral spawns that we had about 6 months ago. So each one of these vials, you can just see those tiny, the little bit of yellow at the bottom. These are a different stage of development of the embryo of the coral. So once we've taken the eggs and sperm from the coral, from the spawning and the research systems literally just up the stairs from here, I then mix and match the sperm and eggs from different colonies. If fertilization is successful, we start seeing the cells dividing, and that's the first stage of what's called embryogenesis, embryo development. And so what I've done for this study is take different stages of development from the moment of fertilization right the way through about a 4-day period until you get something called the planula larvae. And the larvae, this would all be happening in the ocean right up at the water surface. So once the corals are spawned, all of that's floating up to the surface. Those cells are dividing as the embryos developing, and then at the end of that period, this larvae is formed. And at that point, the larvae then swims down to the reef, settles on the reef, and then grows into the primary polyp. And then that divides, and then forms the coral colony that we all keep in our aquarium. So this study, I'm looking at the different cell stages of that embryogenesis, that embryo development. And what I'm doing today is just preparing these samples ready to use under a couple of different microscope techniques. So I'll be traveling across London to use the microscopes at another museum next week. Hey, so I'm now here the other side of London at the Natural History Museum, and what I've got here is the coral samples that I showed you back at the Horniman, those embryo samples that were collected during the coral spawn last year. And what I'm doing over here is now getting them ready to use under the electron microscope. So this piece of equipment behind me here is something called a critical point dryer, and that's going to suck all of the moisture out of those embryos, so we're just left with the sort of dry material, which are then ready to process and slowly get ready for the electron microscope. And that's what I'm going to show you in a minute. So now, following the critical point drying, all of the embryos now are in these little white vessels. They're completely dehydrated, got no liquid left in them. What I've got to now do is, using some really delicate forceps here, I've got to pick up every embryo and then just put it around a little sticky pad on this little stub here. And then the next stage in the process is to coat those with gold and palladium in a process that I'll show you in a minute. This is a really delicate stage of the operation, because I don't want to damage any of those embryos, because we're going to be looking under the electron microscope. Any marks that I make using those forceps are going to show up really clearly. So this takes a long time, looking down through this microscope and just delicately placing them in a circle around the outside of this stub. So out of the critical point dryer, I've mounted those stubs I just showed you. Now we're coating it with this machine, and we're coating to Coming up now, this shows the number of nanometers. We're going to 20 nanometers, and that was coating it with a gold and palladium coating, and that will ensure that when we put it into the electron microscope, we can actually fire the electrons and observe an image. Part of this process uses argon gas, so the argon gas gets fed into this system, creates a pressure and ionizes, creates a vacuum but also ionizes in there, and that allows the gold and palladium to stick to those little tiny coral embryos. So the process is finished now, and now into the electron microscope they go.
Just on the screen here, this is an embryo now that's about 1 day old. So it's going through all the different cellular stages. So what I've done is documented all the fertilization right the way through the points. So I don't know if you can see on this screen here, this slight indentation here, that's ultimately going to become the mouth of the polyp about 3 days after this image. So man, it's an amazing piece of kit. If I just show you, come into this point, and we've got a zooming in, so we can zoom right in on these structures. So if we look here, we can just focus down. Yeah, so this will ultimately be the mouth of the polyp right there, and this is 20 micrometers just there, that scale. So we're looking, you know, pretty close up with that. It's an amazing bit of kit, but and that's it. So I'm here for another day just to document 3 different species right the way through that embryo process.
So the purpose of this is just to document it. What is it relevant wise to your research?
Yeah, I mean, the main purpose behind it is so we now can spawn the corals, but what we want to do is make sure that is there anything in the sort of preconditioning and getting the corals fit for spawning that's disadvantaging the embryo. So what I want to do is check that the embryos are developing correctly, so that, you know, this process of spawning corals in captivity can then be rolled out to lots of different practices, from research to restoration, and then potentially for the aquarium trade as well, developing new color strains of corals for the aquarium trade. That's the idea.
How do you know that they're developing correctly?
So work has been done looking at, this is something called embryogenesis, so there is work that I can refer to in the field. I've also documented this in the field as well as part of the C-Core workshops, so I kind of know what stage they should be at based on some of that field knowledge.
Okay, so you've actually seen it before in prior research in wild coral.
Yeah.
How the embryos are supposed to develop.
Yep.
And now you're checking to make sure what you're doing in captivity is going to match that development process.
Absolutely, yeah.
Very cool. Very cool. All right, well, thanks for the update then. I guess we'll leave you to it.
No problem. And like I say, we'll splice in some of those other videos that goes into it a lot more in depth. So hopefully that will help.
Oh, absolutely. No, it looks fascinating. So that in the background is one of your embryonic corals right now.
Absolutely, yeah.
Very cool. Awesome.
Cool.
Well, I'll catch up with you again next week then. I'm sure you'll have a lot more.
Good speaks, Jay.
Take care, Jamie.
Cheers
Thanks
now.
very much.